so far. Okay, and I wanted to start back here with biofilms because I didn't finish everything I wanted to say about biofilms last time. And remember that they are a large, fairly large and complex and diverse gunk, G-U-N-K, gunk, of different microbes, and they're making polysaccharide capsule around themselves. And the bigger they get, the more diversity they tend to have. And in this picture, you can kind of see that the biofilm is tall, and this part here is showing that it can release new microbes that go off and colonize in a new place. So you can kind of think of a biofilm as a stubborn reservoir, of microbes that both persist in place and seed or initiate new infections. Biofilms can be in your toilet ring, they can be in your mouth as plaque, they can be on rocks in a stream. So like when you've been wading in a beautiful mountain stream and you step on a rock and it's slick, some of that slickness is biofilm, and that could be having algae, bacteria, all different kinds of microbes. So biofilms are the most common way that microbes live. But for human health, and I guess also for pet health, biofilms are a great concern because they tend to form on implanted objects in the body. Yeah, they tend to form on what I'll just list here as body implants. Not so much like breast implants, but things like artificial joints. Okay, people get a hip replacement and they have the artificial plastic or polymer pieces in it. Those tend to form biofilms more than normal human tissue. Things like heart valve replacement. So just put valves, joints, catheters and tubes of any kind that go into the body. If they stay there, as soon as they enter, they tend to form biofilms. And these things, once biofilms form on them, you can't really scrub them. If it's a catheter or like a trach tube or something like that, you'll take it out and put a new one because those are replaceable. But you can't really do that with new heart valves or new surgical joints or stents. Like if you put a stent in a blocked artery. So there's not a good way to scrub biofilm off of this stuff, and there's not a good way to remove it because you can put antibiotics into this system or some kind of cleaning chemicals, but it doesn't make it past that top layer of gunk, and then the bottom is still just living happily on. So controlling biofilms in implants is a huge concern for the industry. And there are a lot of bioengineers working on how can we make these substances be more resistant to biofilms so that they don't form them in the first place. It's really invasive to have to go in and replace those things if they're infected with biofilms. And then those of you going into dentistry, like you'll be thinking about biofilms every day with plaque. At least you can get to that and brush it, or I don't know what it's called when they get the more powerful thing in your mouth. And like for people who haven't been to the dentist in a while, that's at least external and easier to get off. So even if it's this serious or more, like with people that have meth mouth, it's external and you can get it off. So what we're going to return to next, going through the different organ systems or different locations where we have flora, and talk about some highlighted ones of like what are the most important flora in digestive system. So this is showing, except for up in the mouth. So they don't have the mouth on here, but what would be the path that if you ingest food and drink and it's going through your digestive system, what are all the different organs and what are the organs doing? And what is their pH, which is cool. So our digestive system, anything that we ingest through the mouth has to go through pH 2 if it's going to live and persist. And that's a pretty strong barrier against getting infected, to go through that much acid and still live. So ingested microbes are going to be largely killed by stomach acid. But not all of them. So there are microbes that even live in the stomach and prefer pH 2, and they would be acidophiles. And one that you should highlight out of this list from the stomach is Helicobacter. Helicobacter, and that one has involvement with stomach ulcers. Helicobacter is involved with stomach ulcers. It used to be thought that stomach ulcers were just people being really stressed and anxious, but there's an infective component to it from this bacterium that can live at pH 2. Most of the other hugely important gut bacteria are going to be found in the intestines, because that's a much more hospitable place pH-wise. So you're up in the small intestine, you're back up to pH 4 to 5. And in the large intestine, you're back more toward a neutral pH, and lots more microbes can live there. So this group together, of everything that's living, stomach down, we would call those gut bacteria. Collectively, they're gut bacteria. And they, as a group, are much more helpful than harmful. So they are aiding digestion and aiding some vitamin production, especially B vitamins and vitamin K. Your gut bacteria are very important for those. And a lot of those are gram-negative. So the structure of gram-negatives makes them a little bit more suited for a gut environment. And you'll notice in here that we've got several of the ones that we studied in lab. So as a group, Enterobacteria, basically from here down, everything under enterobacteria is classified as being in the enterobacteria big group. So that's got Klebsiella, Escherichia can be normal in the gut. Staph and Strep also normal in the gut. Proteus as well. And then the last thing to write about that is that if you're a microbe and you're going to live in the gut, you need to be BIOL tolerant. So there's many gram negatives in the gut, and whether the microbes are gram negative or not, they need to be BIOL tolerant because there is bile basically from here down in the lower GI tract, there's bile coming out of the liver. So they need to be bile tolerant. Okay. And then we come to respiratory system and what types of microbes we tend to have in the respiratory system. So basically from here, if you kind of draw a line right here, like just slightly below the guy's neck, that's the basic dividing line between upper and lower respiratory. So basically, once you're in the trachea, that's considered lower respiratory with trachea, bronchi, and then lung tissue. And then upper respiratory is anything above the trachea. Make a note here that lower respiratory area in a healthy person should have much lower levels of colonization. If you have large amounts of microbial life in your lower respiratory system, it's usually a problem and associated with disease. That's not true for upper respiratory because if you think about where this is in relation to the outside, upper respiratory is directly connected to outside air. We breathe in an incredible amount of stuff on a daily basis, less so now that we're wearing masks. But your upper respiratory system is dealing with fungal spores and all kinds of dust and debris from the outside world with all different kinds of microbes. So there's much more colonization here than down here. And we have a different set of defenses down here that we'll talk about later. But some of the most important respiratory associated flora are all different kinds of streptococcus, tend to like the environment of upper respiratory. And since upper respiratory is connected with the mouth too, there's a lot of overlap with oral, mouth-based flora and upper respiratory because it's all connected. So we can put streptococcus and other oral flora. Still have yeast, which is also part of the oral flora. And mostly leave it like that. But streptococcus are very common in upper respiratory of all different kinds. Both pathogenic ones and harmless ones. And we could also put for upper respiratory that there's a large variety of viruses that do well in upper respiratory because they haven't passed through very, very many good body defenses yet in that area. So viruses as flora of human bodies, upper respiratory is probably their best or their most prevalent area. Okay. And then we come to urogenital area. So you could refer to this general anatomy, female and male, as urogenital. And there are some big differences between female and male flora because of their anatomy. So for both sexes, male and female, the external microbes are going to be similar to gut microbes. So it's a largely overlapping set as to what you have in colon versus what you have on external skin. Just because you know, hopefully you defecate on a pretty regular basis, and the richness of the gut flora adds to the richness of external genital region flora as well. There will be more yeast on the outside as a proportion of the microbes than on the inside. And probably if there's one thing that I'm like suddenly talking about now and not before, it's yeast. Yeast is pretty normal all over your body. You just don't want it to overgrow. So external microbes are very similar to the internal, but there's more prevalence of yeast. In females, they have some very important vaginal flora that include lactobacillus or lactobacilli. And they are actually really important for reproductive health. Let me move this over here so you can see. The lactobacilli are named that because they're fermenting lactose into acid. And the acid is going to be very important in maintaining vaginal pH. Healthy vaginas should be acidic because of the flora that live there, producing acid as a waste product. So having lactobacilli in the vagina is not a bad thing, it's a good thing. That's the healthy situation. And disruption of that can cause secondary infection. We'll talk about that later, but lactobacilli and other acid-producing bacteria are really important for vaginal health. And probably one final thing to write about this before we move on to pathogenesis. Oh yeah. Is this, that urinary tract infections their most normal cause is when you have a displacement of gut bacteria. So what it takes to get a urinary tract infection is to have bacteria make it to the bladder and colonize there. So in Women, that's a 4 centimeter. No, it's not even that long. Wait, 4 centimeter trip for external bacteria to make it to the bladder. In men, it's about 20. So much more difficult for bacteria to go against the flow for 20 centimeters and make a home in the bladder and cause urinary tract infection than for women when it's just a very short distance. But the main bacteria that cause urinary tract infections are coming from this list. So Proteus is there, and Klebsiella, and Escherichia coli, like we learned in lab. They're not special urinary bacteria. They just happen to come from the gut and be on external genitalia and then make it up into the bladder, which is easier on females. So those are more common in females due to their anatomy, not their hygiene. Not their hygiene. So if you end up working in human health and you have older patients, like you work with geriatrics or nursing home patients, as people get older and they have urinary issues, they tend to get more urinary tract infections in men and women than they would if they were young. Okay, so that kind of ends the section on normal flora. And what we're going to, the section that we're kind of moving into next is about what does it take for a microbe to colonize, and not just colonize, but to end up causing a problem in the people, to cause tissue damage and disease. And your prep video for today was basically me going through this and talking about different parts of it, about what it takes and what are the names of the steps. So what I want to spend most of the time on today is the aspects of pathogens that actually let them do harm. What is making pathogens harmful, or what is making them virulent? So I want to start with this term virulent and its other versions of that root word, virulent. And you might circle on, if you've got this picture, circle the word virulence factors. And maybe make a note that up until that point, so if you go past this point, we're talking about a pathogen, something damaging and it's going to hurt health. This segment here, getting to the point of colonizing and growing, that can be done by flora that are not harmful. So all of this first 4 steps could be done by flora. The extra steps that cause damage, that's what turns something into a pathogen. And if they're going to be pathogenic and harmful, it's because they have virulence factors. Okay, so this word, virulent, equates to the concept of being damaging. So, you know, if we talked about how virulent is SARS-CoV-2 compared to influenza, it's going to depend on age, actually. So like in older people, SARS-CoV-2 is much more virulent than influenza. In children, influenza happens to be more virulent than SARS-CoV-2. So being virulent is like being damaging at some level. And you can compare between different strains. How bad is it? How virulent is it? Virulence is the adjective. Virulence is the noun form of the quality of being damaging. So virulence is a quality. Virulence is the noun quality. Virulent is an adjective description, meaning how damaging. And then the other important term that we need to know to start is virulence factor. Virulence factor. So the more virulence factors that a microbe has, the more damaging it's going to be, or the more virulent it's going to be, or it will have a higher virulence. So a virulence factor is going to be some kind of physical quality, and it's controlled by genetics of that organism, or genetics of the microbe. That contributes to host damage. It's something that the microbe has and that it makes, and a physical property of its being that it has genes for, that makes it more damaging. So for example, the E. coli strain that we use in the lab is called, what's it called? It's the one that we, Alex, what's the one that you use in the worm lab?
I think the like N22 or OP50.
What was that second one that you said?
OP50.
I'm gonna call this one OP50. OP50. So this year in lab, I used the E. coli that they used to feed the worms in the POTS lab. That is E. coli, and it has all of the main essential qualities of E. coli, like being lactose fermenter and not making urease and things like that. There's another version of E. coli, O157H7, that is a very virulent strain that causes food poisoning. It's like one of the famous, possibly deadly, definitely going to make you sick versions of E. coli that sometimes pops up in food poisoning cases. These are mostly the same, except this version has some extra virulence factors that this version of E. coli that we use in the lab, and we could drink it and it wouldn't make us sick. They are basically the same kind of organism, but this particular strain that causes food poisoning has extra virulence factors. And that's what's making it more deadly than the original. And sometimes that can just be 3 to 5 genes. Or the other example is in Griffith's experiment. The S strain versus the R strain. The S strain was the most virulent because it had what that was extra. Remember, Griffith was using those streptococcus strains and infecting the mice. and there was a smooth and a rough. The smooth was deadly because it had what key thing? The protective covering and the capsule. Yeah. And that's the main difference. It's just that one thing. So having the capsule made the S strain more virulent because that's helping it damage the host more. Oh, I know, yeah. So virulence factors can be a wide variety of stuff. But when I'm thinking about what they are and what they act like, it's like weapons of some kind. It's similar to a weapon, and it's something that makes you more dangerous to the host. And what we're going to go through next is what are the different categories of virulence factors and how do they generally get used as a weapon because they're assisting something in this process. They're assisting one of the infection steps and leading to more tissue damage, disease, or suffering in the host. This picture was kind of covered in the prep video, so I'm not going to talk about portals of entry really. And this particular picture is showing a numerical example of the concept that I talked about here with the lab E. coli and the more virulent food poisoning strain of E. coli. But this is comparing two different strains of bacteria, Strep pneumoniae in a highly virulent version. So our lab version is not, because that would be irresponsible of me, versus a moderately virulent Salmonella typhimurium strain. And it's showing down here, if you are injecting these two strains into mice, how many cells does it take of this strain versus this strain to kill a mouse? And so where you see each of the bars, that's a separate experiment where they're taking this many. So 10, 100, and this would be like 75, this would be 125, and this is something like 50. And once you get up to about 125 cells of Strep pneumoniae in the virulent version, which would have the capsule, that's going to reliably kill mice. But if you do the same thing with Salmonella typhimurium, it takes 1 million to reliably kill a mouse. And this type of measurement and this concept is called infectious dose. Infectious dose. And we can define an infectious dose as number of microbes, like that could be cells or viruses or fungal spores, perhaps. Number of microbes it takes to kill the average healthy host. That's infectious dose. And this chart is showing what is the range of infectious dose numbers from some selected diseases. So it's saying what is the name of the disease here, about what is the infectious dose, and then what is the portal of entry. So where they're calling primary route of infection, that's basically like the portal of entry and how you contract that. So measles, it's 1. That is terrible. measles is super, super contagious because you can just breathe in and get established one virus and get the infection and then have all of the suffering that comes from that. Q fever is a bacterial disease, and it's from 1 to 10 cells, and you breathe it in. We see that smallpox is also a small number of viruses it takes to breathe it in and get infected. Make a note here for SARS-CoV-2. The infectious dose is unknown. There are experiments going on right now to determine that. But it's probably going to be not less than 1,000. So it is not as low of an infectious dose as some of these other viruses before. And that probably explains the differences why, one of the differences, like why would one person get it and the other one did not, even if they did all the same things. So, and then having masks, even if you don't block all of the pathogen, you may be blocking enough of it that you're not going to suffer a bad infection. These ones, typhoid and cholera, they are ingested. Why do you think it takes so many cells to establish an infection through ingestion? Yeah, because if they're going to actually cause a problem, it's going to be down in the intestines. That's where these establish a harmful infection, is in the lower GI. And most of the ones that get ingested don't make it through that stomach acid barrier. So it matters, like how do you get exposed and how much got into you and what does it have to get past for you to actually get an infection. That concept of infectious dose is a big deal. Likewise, I know during the early days of this pandemic, we were very more concerned about microbes on surfaces. That is less of a concern now, because having your hands on a surface and then possibly ingesting the virus, this is not surprising. It's a respiratory virus. It's not a GI virus. So on surfaces is not as big of a problem as just breathing it in through the air. But that number probably will become known in the next 6 months, I would think. Okay, this is where I want to have the break. And when we come back from the break, we'll be going through different specific styles of virulence factors. The more virulence factors you have, the lower the infectious dose number is generally going to be. That would be the last fact, really, before the break. The more virulence factors you have is going to lower the infectious dose number. Okay, I'm standing there like thinking, should I go to the bathroom? All right, virulence factors are probably my favorite dork out topic in all of biology. And when my PhD work was actually on a particular virulence factor that encouraged plant infections. And as soon as I learned about the concept, I was just like, that is the coolest thing ever. So we can have different categories of virulence factors, and the first category is any kind of protein molecule factor that a microbe has that is encoded in its genes that allows it to stick and attach on the host and not get swept away. So anything that avoids the microbes getting swept away in the host is going to be considered a virulence factor. So Fimbriae, you can just circle. I think I'll have you circle certain ones on the pictures. Fimbriae, with just the general function of helping microbes stick to a surface, those are considered a virulence factor. because it makes it harder for our body to get rid of those microbes. Kind of like if you walk through a field and you get those cockleburr things or whatever you call them stuck to you and they're hard to get off, fimbriae themselves are considered a virulence factor. Capsules, like we've seen with Streptococcus pneumoniae, capsules are both attaching, pretend I'm writing this right here. Capsules are like a double-edged virulence weapon because they allow both attach and hide. So capsules, we're going to see that on an upcoming slide. In this picture, they're shown as an attachment aid. So for example, mouth bacteria that make a thicker capsule are going to be more likely to contribute to plaque than mouth bacteria that make a thinner capsule, just because they're able to stick on better and resist getting brushed off or washed off with saliva. And then, of course, for viruses, spikes, and having the ability to have a spike match the host receptor and stick on and attach, spikes are considered a virulence factor. You might make a note here on the spikes, as long as they can bind a particular host receptor. If the spike doesn't match anything on the host to bind, it's not a virulent factor. It's not a virulence factor on that host. There's got to be a match. But if there is a match, that virus's spike in that host is definitely a virulence factor. And then, like with SARS-CoV-2 right now, where the spikes are changing a little bit, they're going through the genetic drift, and they bind a little bit better. That's making the strains more virulent with some of those variants. So they either are more contagious or they cause worse disease in people overall because the spike has become better at matching the host. So all of those would be considered virulence factors, or anything else that aids attachment or prevents getting washed away. That's one category. The second category of virulence factors has to do with molecules, proteins, whatever they are, that help the pathogen avoid the host immune system. And to understand this, I need to say a little bit about host immune system, which is what this slide is about. So this slide with the lady and all the words, label this innate, I-N-N-A-T-E, innate immune defenses. These are the type of immune defenses that we have that we don't have to experience the pathogen and build immunity to it. We've always got these things helping to protect us. So these are the style of defenses that are weak, but basically we've always got these to help try and prevent infection. And these are kind of chemicals or structures or processes that are near the portals of entry, kind of as like first responders to help block microbes from colonizing us. So when you breathe in air, it goes into your respiratory tract and it gets stuck in mucus. Well, like the particles in the air get stuck in mucus, and then you have these little cilia cells that flap the mucus back up and out. So every time you like sneeze, you're sneezing out whatever just got trapped in your respiratory mucus. That's a defense that you always have. It's not the strongest defense, but it is something that microbes have to get past. Your skin, just by being continuous and making certain chemicals, it's hard for microbes to get past the skin and get deeper in your body. We talked about stomach acid. We talked about pH changes that are going through here. Your saliva, you add saliva on here. Saliva and tears have a chemical called lysozyme. And the last time we talked about lysozyme, it was a while ago, but lysozyme breaks apart peptidoglycan in bacteria and disrupts their cell walls. So if you get gram-positive bacteria in your mouth and they don't have a capsule, your saliva starts breaking apart their walls immediately and helps protect you. This one, what they're calling flushing, flushing of your urinary tract. Some of us just went and did that during the break. Every time you urinate, anything that's in your urethra is basically going to get washed away in a dramatic flood, unless it's got a way to cling on. So some of those urinary tract UTI-causing bacteria have structures that help them cling on, and or they're able to swim against the tide. So flushing helps prevent colonization. And then the last one on here are these two here about the normal flora. And I want to make a separate note about that. Normal flora are not a virulence factor. I'm put it over here. Normal flora, wherever they are in the body, they tend to live in biofilms. And they're going to be colonizing those surfaces that are near the outside of the body. And we have normal flora basically at every portal of entry. That's one reason I like to go through normal flora first and where they're found. They're living on the surfaces where other microbes have to pass by to get to infect us. And having good, healthy amounts of normal flora at those spots take up space and nutrients, and also attachment sites, and keep those from being available for new microbes that might be trying to colonize us. So your normal flora, that's worth writing down. They're going to take up space, they're going to take up nutrients, and they're going to take up attachment sites at your portals of entry, and there's just less opportunity for harmful microbes to barge in on that, so to speak. So having good healthy skin flora, or good healthy vaginal flora, or your normal colonization of oral and upper respiratory flora is going to prevent other microbes from getting a foothold in those places. So now that we know more about how normal flora act, they are considered part of our innate immune defenses. So if we're going to have colonization, where'd that one go? If we're going to have colonization and growth of a new microbe coming into the body, it's going to compete with flora to establish its living spot. And then it's going to have to also get past all of those immune mechanisms on that slide. Whoops, wrong way. it has to get past all of this stuff if it's going to colonize. So now, knowing that, we can talk about what are some specific ways that microbes get around the immune defenses. And some of these, we've actually seen it in lab, or you've tested for it in lab, but we didn't really say as much about what they're doing in the body. So the first category is if an organism has enzymes that compromise a barrier. This picture that's saying exoenzymes, part A, is demonstrating that concept. So an example of that is certain Staphylococcus strains, particularly Staphylococcus aureus strains, make an enzyme called collagenase, and it breaks bonds in your skin collagen, and that helps it get past the barrier of your skin and infect deeper tissues. That's one example in that category. Another example of evading host defenses in a different way are making toxins that do that same thing. And we should say, what's the difference between a toxin and an enzyme? Alex, do you know this because of the toxicology meetings and stuff? Okay, I just thought. The difference between a toxin and an enzyme is that a toxin is not an enzyme. It's kind of a lame definition, but an enzyme is going to have an active site that enables some kind of catalysis. Like collagenase is an enzyme and it has an active site that binds collagen and then it cuts the collagen. It's an enzyme. There are certain harmful molecules that can cause damage in the body even though they don't have an enzymatic function. So a toxin, we could say that it's a harmful molecule without an enzymatic function. And normally a toxin is going to bind on to something, but not actually change the thing it binds. It's usually a bind and block kind of chemical. There are certain toxins that bacteria make that through an action that's not enzymatic, they can cause your skin to fall apart. So like, have you ever heard of flesh-eating bacteria? Those tend to make toxins that cause your skin to fall apart, and they're not even having an enzymatic function. So the biggest fine distinction, toxin versus enzyme, but we'll see different examples of toxins on these pages in addition to enzymes. Here we have, again, capsule. There's the capsule. So structures that aid in hiding from the immune system or covering up molecules or antigens that are recognized by the immune system. That's a virulence factor. We have specialized immune cells called phagocytes, like cell eater phagocyte, that can recognize molecules on the surface of many different pathogens and say, aha, I see peptidoglycan, that means bacteria, I need to destroy it. But if the pathogen has a covering over it or something that's hiding it from the immune system, nothing to see here, move on, and then it has a chance to colonize the body. So a capsule would be an example of a hide from the immune system, your pathogen shapes. And then this category here, that's shown all across the bottom of this page, is describing something called invasion factors. Invasion factors are going to allow entry into host cells by bacteria. So this is a different way from what viruses do. Viruses can attach with their spike, then they go into the host cell. There are certain bacteria that can make what's called invasion factors that trick the host cell into taking in that bacterium. So they've kind of got a brief example here. Salmonella binds onto your gut lining, first by fimbriae, so that prevents it getting washed away. And then Salmonella will make a pilus. So they're calling it secretion system here. It's a weapon pilus. And it will inject some virulence factors into this gut cell. And those cause the gut cell to start growing a little bit around that Salmonella. And then it tricks the host cell in your gut lining to endocytosing the bacteria. So now it's like hiding on the inside of your body. And it will multiply in your cells, and then it breaks out and goes and infects more cells. This style, you could make a note here, that style of invasion factor, Salmonella does that, E. coli does that in the pathogenic forms. And then plague, like back in the 1300s, Black Plague, that bacterium does this. Salmonella, E. coli, and plague make invasion factors, and they trick the host cell into be like, yeah, come on in, infect me. Also, the plant bacteria that I studied in graduate school, it does that too. They are related species. So all of those things, invasion factors, exoenzymes, toxins, capsules, those would be virulence factors. This I'm gonna, well, I'll come back to that at the end of class. So what we're going to talk about here more toward the end is virulence factors, some specific ones that are kind of famous. So you could call this section famous toxins in human disease. We'll see some specific examples. And we can divide the toxins into two styles. They are either exotoxins, so we're going to consider these categories of toxins. All of them are not having an enzymatic function. They're just binding, they're binding something in the host, and that's causing damage in some way. So we can divide them into exotoxins versus endotoxin. And since we have already mentioned endotoxin before, I'm going to start there. So here's endotoxin. That is just one specific thing. So endotoxin, remember, is broken off lipopolysaccharide from gram-negative outer membranes. And we talked about that is it can cause endotoxic shock if the outer membrane of a gram-negative is falling apart and you've got pieces that break off and float around. The problem with that is it causes a massive immune reaction. And you have body-wide inflammation, and that's what is likely to kill you, not directly the infection itself. Well, not the direct action of the microbe itself. So that's specifically endotoxin. And endotoxin has to be broken off of the microbe. It has to come off of the microbe as it's breaking apart. Exotoxins are something different. Exotoxins are going to be chemicals, molecules, that are going to be released by the microbe into its environment on purpose. They would be encoded in the genome for how to make this certain toxin. And when the microbe senses that conditions are appropriate and that it's in the host, and this would be a good time to like start the war against the host, they will make the exotoxins, and then they will ship it outside the cell and they release it into the environment. So it's going to float to a place in the host and do something damaging. That's exotoxin. And there are lots of different kinds of exotoxins. There's really just one thing that we consider endotoxin. So some of the most famous exotoxins, if you turn the light, it's like gone the whole hour, turn the light off. One of the most famous exotoxins is hemolysin. Hemolysin, as in doing hemolysis. So your strains in lab that did beta hemolysis, that's a toxin that's doing that. It's not actually an enzyme, but the bacteria will sense that they're in a blood environment, and then they start making their hemolysin. This is how you spell that. It's hemolysin. And then they release that into the environment, and it causes the blood cells to fall apart. And what the bacteria get out of this is nutrients, and especially iron, out of the blood cells that are falling apart. They don't make hemolysins all the time. It's only when they sense they're around blood, and it would be beneficial to do that. So you turn the light half on again. Hemolysins are an exotoxin. There's one called alpha toxin. Alpha toxin that specifically, it's not an enzyme, it just forms a pore or a hole in the host cell membrane. So we're looking here, this is cytoplasmic membrane of the host cell. You're right here, host cell. And some toxins that are made by particular microbes, they're not enzymes, but they just build a tunnel through the membrane of the host cell. And then, you know, you've got exchange of outside and inside, and the host cell is lysed. And here comes a bunch of nutrients out into the environment. That's beneficial if you're a microbe out here in the environment and you can get a nearby cell to just lay some nutrients on you. That's alpha toxin. I don't know why that slide is in there twice. Another one that's pretty famous is called diphtheria toxin. So the childhood vaccines where you get different ones at different times and they're for multiple diseases. One of those is against the disease called diphtheria, and the damaging part of diphtheria is that it makes this toxin they're depicting as AB. The B part binds the receptor. The A part comes into the host cell. So again, this is the host cell membrane. And normally, when the host cell is not around diphtheria toxin, it will have its ribosomes and it's building host proteins in the normal way and going about its business. The A section of diphtheria toxin, we can summarize as this. Diphtheria toxin interferes with ribosome action in the host. Okay, so diphtheria toxin makes its way into the host cell and it binds with one of the elongation factors needed for translation. And it's stuck on there, and that elongation factor is basically going to halt the action of ribosomes in the host cell. So host cells that are around diphtheria toxins, they can't produce new proteins, and the host cells are therefore going to die. Let's see here. Yeah, they don't have diphtheria on the infectious dose disease, but diphtheria is a respiratory bacterial infection, and breathe it in. The host cells that specifically have this happen are in the respiratory lining and down in the lungs. So it's especially dangerous for little kids or infants that get this infection in their lung lining, and then their airway lining gets destroyed, and then they can't breathe, and they kind of suffocate in some way from having that infection. It's really sad. We don't have it very much anymore because of the childhood vaccines that kids get. Diphtheria is no longer a problem in the West. Two other really famous toxins that you've heard of before in some way affect the nervous system. And those are botulism toxin and then tetanus toxin on the next slide. Most people today have heard of botulism toxin as being equivalent to Botox, because it is. Botox stands for botulism toxin A. And what that does, so on this picture, the botulism toxin is a triangle. It's actually called botulism toxin A, but this is showing some motor neurons sending signals to a muscle across a synapse here. And so in a normal person, there's a neurotransmitter called acetylcholine that comes out of the neuron. It binds to the muscle, and it will signal the muscle to be able to contract. That's the normal situation. Botulism toxin prevents acetylcholine from coming out of the neuron. So it's binding onto a receptor at the end of this motor neuron and blocking acetylcholine release. And then that's going to cause paralysis. You could write here on the muscle, paralysis or the muscle can't contract. And this comes from a strain called Clostridium botulinum, which is an endospore former. It used to be that at one time people would get botulism is when they would eat food contaminated with the Clostridium botulinum spores, and then spores get into their body and they produce the bacteria, and the bacteria sense that they're in you, and then they make the toxin, and then you get deadly paralysis in your body. Where these days do people tend to get Botox in their body? It's like too many places. Face, like where in your face?
Forehead.
Right here. You're too young to think about it. I've like got that now, like frowny line that makes me look anxious even when I'm not. So here, so that muscle never contracts and you stay undisturbed, no frowny line. Also sometimes here, and you don't want that to drift elsewhere. If you ever decide to get Botox, don't go to the cheapest place. It's got to stay here, not here, because then you can end up with your eyes like that, or the weirdly frozen face. Anyway, the other place where people sometimes get Botox is in their armpits, so that their motor neurons that control sweating stop doing that. So it also can be a treatment for massively over sweating or hyperhidrosis for, I guess, pathological sweat issues can also be used. But it's actually very dangerous, and keeping it in the cosmetic spot is really, really important. I'm just going to look like I look. I don't trust people to get it right. And then the other really famous neurotoxin that's coming out of bacteria and affecting nervous system is tetanus toxin. Tetanus toxin has an opposite action of botulinum toxin. Okay, so this situation is a little bit more complicated. Here's the muscle still. Here's the motor neuron that's supposed to be telling the muscle to contract. So that's the same as what we saw from the previous slide. But there is another set of neurons that are called inhibitory interneurons that will kind of step in and tell the motor neuron, stop sending a contraction signal. So this is a little bit less of a common situation, but they're supposed to be sending glycine amino acids to the motor neuron to block acetylcholine production and then stop the contraction. Tetanus toxin binds and blocks tetanus toxins bind and block inhibitory neurotransmitters like glycine. And they prevent the inhibitory interneuron from telling the motor neuron, okay, that's enough, stop signaling contraction. So the end action of tetanus toxin is causing continued muscle contraction. So in that case, whatever muscle is contracting is going to be locked into contracting. And the old name for tetanus as a disease was lockjaw. Lockjaw. Because one of the places that tended to get contracted and stuck was the masseter muscle. So your mouth is frozen shut. The other places that tetanus can lock muscles are in people's back, and they get stuck like with an extreme back flexion. It's very painful. This problem is caused by a related strain of bacteria that's Clostridium tetani, which is also endospore former. And if you ever step on metal outdoors and something dirty from the outdoors goes up into your foot, you are supposed to be current on your tetanus vaccine so that you don't have this happen. So tetanus vaccine is another one that's in the childhood series to prevent this from happening. If you get older than, like say around 25, and you step on a rusty nail or whatever the thing is, they usually will give you a tetanus booster shot pretty quickly so that you don't have growth of the bacterium. And actually people my age are supposed to get tetanus booster shots when you're around my age, which I need to do. But that's another virulence factor toxin. And that is where we will stop today. So on Tuesday, there will be some other video that you need to watch and then do a quiz on. And we'll have a little bit of, like half a lecture on immune system. And then you'll need to have your group of 3 or 4 established, and then you'll work on a half class length little project for our group work on Tuesday. Okay. So probably what I will do is have, I'll take pictures of the people's projects. So what it is writing on the board, like certain details about a disease, virulence factors in the immune system overcoming. And then I just have, we kind of go around and tour the different ones. So I'll just take pictures of them and send that. Yeah, that would be good.
I should have showed you. I thought we were submitting like a hard copy of the unknowns. I submitted it online. But when you look at it, the gram stands are big. I thought the hard copy you'd have to look at them, so that's why they're so big.
Okay, they're just gonna show up really big.
Not like crazy big, but I mean, they're.
Okay, that's fine.
Just if there's a hard copy, you can see what you're looking at.
This is the first time I've had this class turn them in online. So it's kind of nice, I don't have to carry papers around.
Yeah, that's true.
Stuff.
I just want to let you know.
So for the title
good.
thing, do I just give that to you?
Yeah, this looks good. You will want to kind of look for more articles like this, because these will be good background info. For your actual presentation. You would want to have, maybe this article and one more, or primary sources.
Research.
But no, this looks good, and I'm excited about this topic. I was actually thinking about it at night.
Really?
Last night, I was like, yeah, I'm thinking that'll be a good, anyway, excited to advise this topic. Thank you. I saw your email this morning, and I answered it right before class, but I don't know if you saw it.
Yes. So I can do, let's see, tomorrow. I can do tomorrow. Can I take it in the morning at 8 a.m.?
Yeah, that would be really good,
Yay.
actually. Because I will be here, I'm usually here at 7:35 or something like that.
Perfect.
That would be good.
Do you want me to meet you in Whitehall?
Yeah.
Okay.
Yep,
I can
that
do
would be good.
Thank you for giving me an
Mhm.
extra day. I woke up this morning and I was studying and I didn't feel comfortable and I want to do good, so.
I don't want to leave it too long, because
Right,
I don't want you to, like, let things
pile on.
snowball into finals, if that makes sense.
Right.
Yeah.
Thank you.
Yep. Okay, that would be good. I will be there.
So to get the vaccine, where do I go?
It's just like up there on the oval outside the chapel.
Oh,
They're set up out there. And I think at 9, they're starting with international students, but at 10, it starts up with any students